Let's start off by exploring our sense of taste, or gustation as it's more formally called. A key thing to understand is that what we typically consider to be taste is actually a combination of multiple sensing modalities. Taste arises from the perception of chemicals contained in your mouth when you eat something. The full perceptive taste, though, also depends on the smell, texture, and temperature of whatever you place on your tongue. Additionally, the level of pain induced by something in your mouth, like a jalapeno pepper on your tongue, influences the way it tastes. The four classic axes of taste are sweet, salty, sour, and bitter. Our perception of sweet signals the presence of sugars and starches in our food, which are essential biological macromolecules that nurture us and keep us alive. The perception of salty indicates the presence of sodium and potassium ions, which, as you know, are critical for the proper functioning of our neurons. The taste of sour occurs in the presence of acids. Lastly, bases and toxins trigger the perception of bitterness, preventing us from experiencing negative physiological effects by consuming them. More recently, a fifth taste was added to our classic list: umami. In 1985, the term umami was recognized as a scientific term for describing the effect of tasting glutamate and other nucleotides, most often found in savory food additives such as monosodium glutamate, or MSG. Now, 5 taste modalities might not be the limit. Within the last decade or so, many more tastes have been discovered and are the topic of active investigation. These new tastes include the specific perception of gases, such as carbon dioxide, or CO2, as well as select fatty acids, and even water. Now that we've identified some of the main taste modalities, let's take a look at why we're actually able to consciously taste these things. In other words, let's explore the underlying neurobiology. First, where do we register taste? This is an easy one to answer, since you're probably all familiar with it. The first, most prominent sense of taste and flavor occurs at our tongue. In the surface layers of our tongue, taste receptors are assembled into taste buds, which are packed together into taste papillae. We won't concern ourselves with the details here, but in these taste buds, specialized taste receptor cells form synapses onto gustatory nerve fibers, which project deeper into the brain. Now, you may have heard that specific areas of the tongue are selective for a given taste. While many textbooks would have told you this prior to the year 2000, the science of gustatory sensation is rapidly evolving and has made great progress over the last 10 to 15 years. We know today that while we clearly have different taste receptor cells for each taste, all of these receptor types are distributed reasonably uniformly across the tongue. Let's now take a look at how some of these tastes are actually sensed. We'll start with 2 classic tastes, sour and salt. Both are sensed by ion channels that carry or are activated by specific ions: sodium in the case of a salty taste, and protons in the case of a sour taste. In both cases, receptor activation leads to a positive ion influx into the sensory cell and the subsequent depolarization of the cell, which in turn leads to the release of neurotransmitter and activation of the gustatory fibers projecting to the brain. What about a newer taste, such as carbonation? It turns out that some sour taste receptors express an enzyme on their surface called carbonic anhydrase. Carbonic anhydrase, or Ca, catalyzes the conversion of carbon dioxide and water into bicarbonate and protons. It's thought that the protons then activate the sour receptor and subsequently the nerve fibers going to the brain. So in a nutshell, the taste of carbonation, or CO2, is sour plus fizz. Think about that the next time you enjoy a can of soda. Other newly discovered tastes include our ability to detect fatty acids and even water. Let's start with fat. Humans and select animals taste fat very distinctly from other flavors and textures. You probably experienced that yourself if you've ever enjoyed a fatty but tasty prime rib. Researchers at the Nestle Research Center in Lausanne, Switzerland, recently showed that 2 G-protein coupled receptors, called GPR40 and GPR120, respond to specific fatty acids, and that mice lacking these receptors no longer prefer water-containing fatty acids to plain water. The case for tasting pure water, on the other hand, is less clear. While there's psychophysical evidence that humans can actually taste pure water, there's not yet a molecular explanation for how this happens. However, a receptor that responds to water has recently been identified in flies. This receptor, called PPK28, is an epithelial sodium channel that depolarizes sensory cells in the response to pure water, but not to water with dissolved material that raises its osmolarity. The jury is still out on whether or not such a physiology applies to humans, but it's definitely an interesting lead. Let's now consider the tastes of bitter, sweet, and umami. Pharmacological studies long suggested that bitter, sweet, and umami taste inducing compounds bind to G protein-coupled receptors and activate them. If you recall from our earlier modules, once G protein-coupled receptors are activated, they induce the formation of second messengers, which in this case activate cation channels that depolarize the cell and cause it to release neurotransmitter into the gustatory nerve fibers. Since GPCRs constitute the largest integral membrane protein family in the human genome, with close to 1,000 members, the big question is, which of these receptors are taste receptors? This has been studied extensively, and the story that follows is a remarkable example of combinatorial neurobiology. The search for taste receptors in the human GPCRome led to the identification of several candidate taste receptors, broadly classified as T1R and T2R receptors. Both groups are expressed in taste cells, but they're expressed specifically. That is, many taste cells express either T1R or T2R receptors, and some express neither, but none ever express both at the same time. Curiously, they're also quite related to odorant receptors, which mediate our sense of smell. More on that later. Structurally, while both groups belong to the family of 7 transmembrane domain GPCRs, they have some key differences. T1R receptors, of which there are currently only 3 members identified, possess a rather large extracellular N-terminal domain, which presumably is involved in taste reception. T2Rs, in contrast, don't have this large N-terminal protein domain, but they're far more numerous. To date, over 40 members have been identified that respond to specific molecules. These findings led to the hypothesis that these 2 groups were taste receptors for bitter and sweet, with one class encoding receptors for bitter taste and the other for sweet tastes. Of course, this idea had to be tested using the scientific method, and the results were quite stunning. Experiments using both in vitro and in vivo model systems clearly showed that T2R receptors encode bitter taste reception, while T1R receptors encode sweet and umami tastes. Curiously, while individual T2Rs can detect select bitter compounds, such as T2R10 for the bitter poison strychnine, combinations of these 3 members of the T1R receptor family are required to detect either sweet or umami. When studied in isolation, individual T1R receptors don't seem to produce any neurobiological response. When present in combination, however, T1R2 expressed together with T1R3 in the same cell allowed the cell to perceive sweet compounds, while T1R1 expressed together with T1R3 in the same cell allowed the cell to perceive umami. So far, we've talked about a rather large molecular and chemical compendium describing taste perception. T1R combinations allow us to detect sweet and umami flavors. About 40 T2R receptors can detect bitter compounds. Epithelial cation channels can detect salt, and other mechanisms allow us to detect sour and fizz. In the next lesson, we're going to move beyond the receptors to see the neurobiological pathways that lead us to our perceptions of taste.